Good afternoon and welcome to Cure Talks. This is Priya Menon, your host, joining you from India. And today we are discussing CAR T-cell therapy treatment for cancer. As we all know, FDA has approved the first gene therapy treatment in the US. And our guest for today is a man who led the team behind it all, Dr. Carl June, a pioneer and specialist in T-cell biology and lymphocyte activation at the University of Pennsylvania. The new therapy uses the body's genetically engineered immune system to find and kill cancer cells. In the coming hour, we'll be discussing the nuances of this therapy. On today's panel, we have patient advocates Gary Peterson, Jack Aiello, and Mike Scott from our Cure Talks community. Just brief you on the format that we'll be following today. I will start with a few questions, and then Gary will begin the patient panel discussion with Jack and Mike. Towards the end of the talk, we will be taking in questions from the audience. The dial-in to ask your question live is 718-664-6574. I repeat, 718-664-6574. This number has been posted on curetalks.com webpage as well, and we will be repeating this number before we begin the QA. You can also post your question in the comment section of the Cure Talks page or email me, priya@trilix.com with the same. I once again welcome everyone to today's episode of Cure Talks. Moving on to the discussion, Dr. June, it's such a pleasure to have you here. Welcome. To start off, can you talk a bit about how your research with CAR T cells began and whether your background as a Navy physician has had anything to do with it?
Well, thank you very much, Priya. So I began to study as a physician and then oncology and in the early 1980s, and at that point, the new emerging therapy was a bone marrow transplant. And in the early days of transplant, they thought that it would work because they could give what are called super lethal doses of chemotherapy. So chemotherapy so intense that it would kill you unless you were rescued with bone marrow from a donor, usually a brother or a sister. And and in fact, that was the first time that sometimes leukemia could be cured. And but it was only later when they did a set of identical twins and they gave them the same chemotherapy as they gave transplants who had a donor was a brother or sister. And what they found out was that it only worked with increased survival if you did not have an identical twin as a donor. So what that means now in retrospect is that the differences in the immune system between the brother or sister who was a donor are what led to the long-term remissions in leukemia. And if you had no differences, meaning an identical twin, it wasn't as good. The only benefit you got was a high-dose chemotherapy. So the problem back in the early 1980s is that the procedure often had very severe side effect called graft versus host disease, where the immune system of the brother or sister would attack the recipient patient. And so I began working on how to get around that, and that ended up being, could we do the same benefits of a bone marrow transplant but not have to do the transplant and actually use your own immune system as the weapon? And over about 25 or 30 years now, that's now culminated in an FDA-approved therapy called CAR T cells. So that's, I started off because of that, learning how to grow T cells was the key from the patient. And then, I was in the Navy because of the reason we wanted that bone marrow transplants was, it's been recently shown, we had, was for radiation casualties, as could happen, for instance, in things like when Chernobyl or the Fukushima reactor accidents in Japan. What you die from that radiation exposure is bone marrow failure, and it can be treated with a bone marrow transplant. So in the early 1980s, the Navy paid for my education so that I could learn how to do bone marrow transplants, and then if we ever had an issue, say, with a Navy reactor on a submarine, we would have a potential way to treat the sailors. And so that's how I started on that initial research. But then when I began later, after in the almost early 1990s, what happened was we had the collapse of the Cold War, and really the reactors and so on became a low priority issue. And the only research I could do in the Navy was on HIV and malaria, which are infectious diseases. So the first use we had of giving T cells to patients were actually patients who had HIV and AIDS. And so we showed that we could take the T cells out from a patient who had AIDS, grow them up in the lab with the technology that we developed, and then give them back to the patient, so the same patient. And their T cell counts got much better, so their immune system started to work, whereas it had been failing. And then, so that was throughout the 1990s. And then I retired from the Navy and went to the University of Pennsylvania in 1999, and we began using the same approach, but in patients who had leukemia. And so we used all the technologies we had learned in HIV and then transferred that to cancer patients, and that's led now. Our first patient was treated in 2010, and then now FDA approval just in August of 2017. So in a nutshell, that's how it happened.
Yes. So you just mentioned having worked with HIV and engineering the T cells. Can you talk a little bit more about your work with HIV and why HIV was ideal for this engineering of the genes?
You know, one thing about this is it shows that if you can work in different fields, seemingly disparate fields like an infection and cancer, you can sometimes have lessons or low-hanging fruit that applies in one field to help solve a problem in the other field. So in the cancer field, the problem was, how to make T cells attack your own cancer but not attack your body. And we didn't have an efficient way to genetically alter those T cells. And it turns out HIV has evolved to do exactly that. It infects people and it goes into their immune system, into T cells. But the natural HIV virus, as people know, destroys T cells eventually. And so the field, a number of investigators over the years developed HIV and modified it so it would be a tool to insert genes into T cells rather than kill them. So what they did was basically gut the virus of the genes and material that would allow the virus to kill T cells, but retain the ability to insert new genes into T cells. And so we were the group to first test that in humans. In 2006, we treated patients who had late-stage HIV with their own T cells, but then using this modified form of HIV, which we call a lentivirus, to insert a new gene into their T cells. And then that's what we ended up using in cancer patients because it works with very high efficiency and has been very safe. In hundreds of patients, we've never had a side effect, from this virus.
That's great. So this is a very common question that we have come across as to how this new therapy is different from the standard chemotherapy.
Yeah, it's radically different. It's, for one thing, the therapies that we have had until just recently have, other than surgery, and if you can't cut it out, that has meant then that usually it's either surgery or radiation or a combination. And both of those are non-specific therapies, and they work by killing cells, but they also kill normal cells, and which leads to the side effects. And with an immune therapy that's precisely targeted at just the cancer cells, there aren't those so-called off-target effects. Off-target effects are when the therapy is killing cells, but it's not helping you get better. And so chemotherapy, as most people know, has many side effects from symptoms such as nausea or vomiting to very severe issues that can involve liver failure, heart failure, and bloody diarrhea and so on. Those are off-target effects, and they actually don't help you get better from the tumor. And an on-target effect then is its side effects related to actually when you're killing the tumor, and so those happen when the patient is actually benefiting from the therapy. And the first one we found with our CAR T cells, and CAR, by the way, stands for chimeric antigen receptor. And basically that means it's a T cell that's been modified. The word chimera is used because it's from the Greek a chimeric beast, which is a fusion of two different kinds of animals, in the Greek mythology. So we have as a chimeric T cell. The T cell is a fusion of two cells in the natural immune system, which is the B cell. And B cells normally make antibodies, which we use in our defense against viruses and infections. And the T cell normally just kills cells but does not make antibodies. So a CAR T cell takes the best worlds really of a T cell and a B cell. And the T cell can be a killer cell of tumor cells, but it also now has antibodies in it. And that's how we can direct it to kill tumor cells. And the first on-target effect we found from CAR T cells was very high fevers. And in our first adults, they had fevers of 106 degrees that would last for several days. And there was no infection. And we now call this cytokine release syndrome, or CRS. And it only happens in patients who are getting better. The fever happens when the tumor is when the CAR T cells are killing the tumor cells. And it's a violent reaction. It can last up to a week. We now have ways to manage it. And we've learned a lot about CRS. So as I mentioned, it has high fever. The fevers are usually higher in children than in adults, probably because the children's immune system is more robust. And when we've also learned that if you treat patients earlier when they don't have as much tumor, there's less CRS. So our first patients, the first three that we reported, that were treated in 2010 were adults, and they had, by the scans and so on that were done, they had between 5 and 7 pounds of tumor each, just massive loads of tumor. And when their tumor got ablated by the CAR T cells, it was violent. They had fevers that lasted, in one case, 2 weeks until the tumor was all gone. But then the patients recovered and they're completely fine. There's no long-term side effects from that. So that's an on-target side effect, is cytokine release syndrome. It can be severe in patients with high tumor loads. Up to 1/3 of the patients go to the ICU, but the fatality rate from this is less than 1%. In the trial that Novartis did to get FDA approval of the CAR T cells for leukemia for patients between age 3 and 25, 48% of the patients had cytokine release syndrome, but there were no fatalities.
Thank you, Dr. Arjun. Before I hand over to Gary to start with the patient discussion, can you share some Eureka moments that was part of this journey, this T cell therapy journey?
Yeah, I mean, we started off first with just, could we, and what we're doing really, this kind of CAR T cell therapy it's a new form of transfusion medicine. So the first successful transfusions in humans were blood cells. And, and for that to happen, they had to develop the typing of the blood system, so the so-called ABO antigens, and to find out, who could be a blood donor and so on. And now what we're doing really is T cell, because CAR T cells are a form of blood cells. We're doing T cell infusions. And so our first Eureka was when we actually treated those AIDS patients. We made CAR T cells for HIV. And we gave them a single dose of CAR T cells, and we saw immediately their T cell counts went up. In many cases, they doubled and went to normal levels. And so that was evidence that the CAR T cells could go into the patients and stay and actually survive, and start dividing. And so that was what we were hoping for, and it worked better than we thought it would. But in a really curious twist, the FDA has a rule in the U.S. that patients treated with gene transfer technologies, so this is a form of gene therapy because the cells are genetically modified, and in this case, the patients got CAR T cells for their HIV, starting in 1997, and the FDA has a rule that each patient needs to be followed for up to 15 years, and tested each year to make sure that it's been safe and that there's been no toxic effects from the genetic engineering. So we analyzed in 2012 all the patients we had treated back in 1997 and to 2000, and there were 40 patients, and we analyzed them because the FDA had a rule that if you could show that the gene transfer was gone, in other words, had gone away from the patient, then we would no longer have to have the expense of following the patient every year. So we analyzed all our patients from the samples in this freezer, and we're astonished to find that, in fact, more than a decade later, they were all still engrafted and had these CAR T cells at very stable levels. No one expected that. In the previous trials with transfused T cells like that, they had not ever lasted longer than a week. And we found that the half-life of these T cells is more than 10 years in patients with HIV. So it suggested for the first time that you could make a living drug. You could genetically modify someone's T cells and give them back, and they would then be there on patrol for potentially the rest of that patient's life so that they could act in a way like a vaccine. So that was the first amazing aha moment.
That's
drug.
Exciting.
Yeah, and then the second major one was really in 2010 when we treated the first patients with these CAR T cells. This one was called CTL019 or CART19 because it targets it's a CAR T cell against a molecule called CD19 on leukemia cells. And when we treated those patients, we didn't know what would happen. All previous CAR cell trials in patients with cancer had failed and not shown any effects. And all three patients had remarkable remissions. And it was so unexpected that patient one, who we reported in the New England Journal of Medicine in 2011, patient one, when the bone marrow biopsy came back with no leukemia, I didn't believe it. I said, that basically they must have missed it. And so leukemia is in the bone marrow. Usually they biopsy part of the pelvis, and I just said biopsy the other side. And so we did the first biopsy on day 28, and it was negative. And then the biopsy on day 31 from the other side came back also normal with no leukemia. So that was astonishing, and we had that repeated then many other times.
This is so exciting. Awesome results.